Okay, let's unpack this. We're doing a deep dive into a really fascinating, but I think critically ignored aspect of aging.
Which is the scheduled, almost programmed collapse of our immune system.
Exactly. It's this quiet clock that's just running in the background, making us vulnerable to everything from, the flu to cancer.
And it's all tied to a tiny organ you might not even remember you have, the thymus.
The thymus gland.
It's really the Achilles heel of our longevity. This gland, it's tucked right behind your sternum, and it's responsible for producing your specialized T cells.
The elite soldiers of your immune system.
Right, the highly trained ones. But the thing is, it's programmed to decline. It starts shrinking and shrinking dramatically right after puberty.
Mad early.
Oh yeah, the process is called involution or immunosenescence. By the time most people hit 60, their thymus is, for all intents and purposes, just replaced by fat. It's functionally gone.
The program literally begins when we're teenagers. So, the mission for this deep dive is to explore two radical, very modern approaches that are taking this on directly.
Right. We're looking at these cutting-edge methods trying to manually override that decline to essentially reverse immune aging.
And our sources for this are really exciting because they span the whole clinical spectrum.
They do. We're comparing established human trial data from Dr. Greg Fahy and his famous Fahy Protocol, that's a pharmaceutical cocktail, with data from a completely revolutionary, brand new approach.
The DFI protocol.
The DFI protocol, which uses transient mRNA technology. And this is fresh out of the lab. The paper was published today, December 17th, 2025, in Nature.
So we have this kind of philosophical battle, a sophisticated, almost low-tech pharmaceutical balancing act versus a high-tech, programmable genetic update.
And both are aiming for the same prize.
A youthful immune system. So let's start with the revolutionary thinker who really pioneered the human approach, Dr. Greg Fahy. He based his work on this foundational idea that aging isn't just random wear and tear. It's not just entropy, your body falling apart.
Right. He saw that the thymus follows a specific programmed pattern of decline. Very similar to how, say, a cell undergoes apoptosis or a woman goes through menopause. It's a built-in program.
And that programmed nature is the conceptual key, isn't it?
It's everything. Because if aging were purely random, purely entropic, reversing it would be almost impossible. But Fahy theorized that if the decline is biologically induced, well, it's like a sophisticated piece of plumbing.
You can intentionally stop it up?
Or reverse it. He famously quotes Scotty from Star Trek, the more complicated the plumbing, the easier it is to stop it up.
I love that. So that philosophical starting point, that's what led him to the TRIM protocol.
Yes.
This three-part cocktail. But the goal is simple. Regrow the thymus. You can't just dump the active ingredient into the system, though. There's a serious roadblock.
A huge metabolic roadblock. The primary engine here, the thing that actually regrows the thymus, is growth hormone, or GH.
Okay.
And early animal studies showed it works wonders on the gland. But GH has a massive immediate downside, especially in older people.
And that is?
It drives insulin insensitivity. It basically temporarily blocks how effective your insulin is.
So you're solving an immune aging problem by introducing a massive metabolic aging problem.
Precisely. You would be accelerating cardiovascular disease, atherosclerosis, hypertension, all these pro-aging effects that are the exact opposite of what you want.
Right.
Fahy realized this metabolic penalty was the real hurdle. It forced him to turn what could have been a simple hormone treatment into a really sophisticated metabolic balancing act.
So the rest of the cocktail is basically just a shield against the side effects of the main treatment. What did he add?
The first addition was DHEA. That's dehydroepiandrosterone. It's an adrenal steroid that plummets with age.
Yeah, DHEA.
Fahy hypothesized it might block that diabetogenic effect of GH, and he actually proved it on himself through self-experimentation.
Wow.
But for extra insurance and, for more potency in the older participants, they had one last thing.
Metformin.
Metformin, the ubiquitous anti-diabetic drug.
So they're co-opting a drug that's already known for improving longevity markers to specifically neutralize the side effect of their main thymus regenerating treatment.
That's the brilliance of the combination. Metformin's job here is just to make insulin more effective, directly counteracting the problem caused by the growth hormone.
And it let them regrow the thymus without that systemic metabolic penalty.
Exactly.
And the initial TRIM trial, this was between 2015 and 2017 with, 9 men age 51 to 65.
That's right, a small but significant trial.
And it yielded some really impressive results. Beyond just regenerating the thymus, the objective beta showed an average of a 2.5-year reduction in epigenetic age.
Yes, a clear reversal on a molecular level.
Yeah.
And we also saw improved kidney function, and even that surprisingly visible finding of some darkening of hair color.
Which is anecdotal, sure, but it suggests broader systemic effects. It's not just about T cells.
Absolutely. And there's one other critical element here, Fahy's approach to how it was administered. He called it pulsing.
Pulsing the treatment. The idea being that if this works, the treatment shouldn't be continuous. Why is that so important?
It really speaks to the lifespan of the new immune cells. I mean, if you successfully regrow the thymus and you generate a fresh cohort of these naive T cells, those cells should last for maybe 20 years.
20 years, so you don't need to be on it all the time?
You don't need to continuously bathe the body in high levels of hormones. Pulsing lets you get the benefits, that rush of new T-cell output, before the potential bad stuff kicks in.
Like potential cancer risk or sustained high IGF-1 levels from continuous GH use?
Exactly. You just turn the engine on, generate that immune capital, and then you turn it off until you need another dose, maybe years down the line.
Okay, so if TRIM is the art of pharmaceutical balance, DFI is just the pure power of programmable biology.
A totally different approach.
Let's make that transition and look at this revolutionary new strategy. We're moving from a balancing act of known hormones to precision mRNA technology.
But aiming for the exact same target, immune rejuvenation.
Right. And what's so fascinating here is that the DFI strategy it sidesteps that whole GH metabolic problem entirely.
It does. Instead, the researchers identified these key immune cues signaling pathways for Notch, FLT3L, and IL-7 that are diminished in aged individuals.
And they decided to just transiently reconstitute them.
Exactly. The factors are delta-like ligand 1, or DLL1, which activates Notch signaling, then FLT3L and IL-7.
And instead of trying to physically regrow the thymus with systemic hormones, they just deliver the mRNA for these factors using lipid nanoparticles, or LNPs.
Right. The same LNP technology from the COVID vaccines.
And the technology delivers those instructions specifically to the liver. Why the liver?
Well, the liver, unlike the thymus or most other organs, maintains its incredible protein synthesis capacity even in very advanced age.
So it acts as a temporary factory.
A robust temporary factory. The mRNA is the software update, the LNP is the hardware delivering it, and the liver just reads the instructions and starts pumping out these youthful factors. It's a really clever way to bypass the shrunken, involuted thymus.
I think the contrast with older approaches, like traditional recombinant proteins, is so important here. It really shows how far this platform has come.
Oh, absolutely. What's the core advantage of mRNA versus just injecting the proteins themselves? It comes down to two things: safety and control.
Okay.
Older recombinant proteins, especially cytokines like IL-7, cleared out of the system so fast you had to give frequent, really high doses.
And that caused problems.
Big problems. It always resulted in significant systemic inflammation, which is the last thing you want to induce in an aged or frail person.
You mean a big inflammatory response?
Exactly. The source notes that older approaches caused these marked elevations of inflammatory factors like GM-CSF, IL-10, IL-1 PAR.
But the mRNA approach is different.
Very different. It's transient, it's controlled. The liver produces these sustained levels of the factors for about 48 hours, and it achieves much lower amplitude peaks. For IL-7, they got peaks that were 10-fold lower than the standard approach.
So it's a cleaner, more targeted signal without the systemic immune storm.
You got it.
So what did this highly controlled approach actually achieve in the mice? These were 72-week-old mice, which is pretty old for a mouse.
The effects were robust and definitive. First, DFI partially restored the physical size and the cellularity of the thymus.
So it did help the thymus itself.
It did. But more crucially, it robustly expanded what are called common lymphoid progenitors, or CLPs. Think of these as the factory floor managers that churn out new immune cells.
And those get severely depleted with age.
Right, so you're replenishing the precursor cells.
And what was the measurable result in the bloodstream?
This translated directly to the periphery. DFI increased both the frequency and the absolute number of circulating naive T cells.
Those are the fresh young cells that haven't seen an antigen yet, the ones we lose as we age.
The very same. And critically, it raised the naive-to-memory ratio. That's the gold standard for indicating true, new T-cell output, not just, the tired proliferation of existing old cells.
It's proof of genuine regeneration. Okay, regenerating T cells is a huge scientific achievement, But the functional benefits, that's the real test for any longevity intervention.
Of course.
So let's look at the real-world applicability, starting with, well, the most relevant topic today, vaccine response.
And this is where DFI really shined. They used an established adjuvanted ovalbumin, or OVA, vaccine model in these aged mice.
Right.
The mice that were preconditioned with DFI showed nearly a twofold increase in antigen-specific CD8 plus T cells compared to the control mice.
A twofold increase. That's a dramatic boost, especially considering how poorly aged immune systems normally respond to vaccines.
It is. It's measurable immunological rejuvenation that addresses a major public health concern.
And the source material actually puts a number on this rejuvenation.
It does. It says DFI effectively restored the vaccine response in these aged mice to levels comparable to mice that were approximately 24 weeks younger.
So they rolled back 6 months of immunological time.
With a simple transient intervention.
Yeah. Okay, let's move to cancer immunity. Immune checkpoint blockade, ICI, is one of our most exciting cancer therapies, but its effectiveness often just tanks with age.
Because the immune system is too depleted to launch an attack.
Exactly. So did DFI help older mice respond better to these checkpoint inhibitors?
It showed powerful, game-changing synergy. They used the aggressive B16 OVA melanoma model. Now, anti-PD-L1 ICI treatment alone, it conferred very little benefit in the age control group.
The standard treatment basically failed.
It failed. But when those aged mice were preconditioned with DFI, the combination drove complete tumor rejection in 40% of them.
40% complete rejection where the standard treatment was ineffective. That completely shifts the conversation about treating age-related cancers.
It really does.
What was the mechanism? What was happening inside the tumors themselves?
DFI increased the infiltration of tumor-specific CD8 plus T cells, but significantly, it also increased their clonal diversity.
Meaning the immune system didn't just send more soldiers.
It sent a broader, more diverse army of cells. Cells capable of attacking different aspects of the tumor. And what's more, these T cells showed lower exhaustion markers. They were fresher, more effective fighters.
Exactly what the checkpoint blockade needed to work. But wait, we're talking about aggressively boosting the immune system here. The obvious concern is autoimmunity.
Of course. Are you pushing the system too far?
Right, are we walking a tightrope?
That was the critical safety assessment they performed, and the results were incredibly promising. They tested DFI in 2 established models for autoimmunity.
Okay.
One for type 1 diabetes and the EAE model, which mimics MS. And DFI did not exacerbate either disease. It didn't break self-tolerance. The immune system didn't suddenly start attacking the host's own tissues.
And because it's an mRNA system, the effects are controllable. That adds a safety margin you don't get with long-term drugs.
That transience is a huge safety advantage. The immune boost is largely confined to the dosing window, about 28 days.
And then it reverses.
It largely reverses upon withdrawal. So if an adverse event were to occur, you can stop the treatment. The effect isn't compounding over decades, like continuous hormone use might. So when you look at these 2 solutions together, you start to see just how powerful the future of immune rejuvenation is.
2 very different paths to the same goal.
Right. TRIM is this clever pharmaceutical approach. It relies on carefully balancing growth hormone's power with its metabolic side effects. And it's applicable today. We have human trial data.
With the promise of very long-term benefits from short, smart pulsing.
And DFI is the clean, programmable future. It's an mRNA platform that uses the liver as an outsourced factory to transiently restore specific factors.
Showing these incredibly strong functional results in mice vaccine response, cancer response, without the inflammation that plagued older methods.
But the underlying goal of both, it's the same. It's immune system reprogramming. And Fahy's broader vision shows just how profound this could be. It goes way beyond just treating aging.
You're talking about his work in cryopreservation and organ bank?
I am. It sounds like science fiction, but the source makes it clear this is a logical extension of what we're talking about.
Explain that.
Fahy's lab is heavily invested in this idea of putting a foreign organ, say a kidney, into a cryobank for a few months.
Okay.
And during that time, the recipient's immune system, the thymus specifically, is reprogrammed using these regenerative techniques to accept that foreign organ as self.
Which would eliminate the need for immunosuppressants for, what, 25 years or more post-transplant?
That's the goal, a massive clinical shift. It means reprogramming the immune system isn't just about longevity. It's about overriding the body's hardwired rejection mechanisms.
And the source mentions this could also be used to eliminate autoimmune diseases.
Potentially over 80 different autoimmune diseases. They've already shown it can reverse established autoimmune diabetes in animal models. So the core concept, reprogramming the immune system, whether it's with a drug cocktail or mRNA, that is the foundational element that enables all of this. It proves that aging isn't necessarily a failure of regeneration, but a failure of instruction.
Which brings us to our final takeaway. The thymus isn't just a passive indicator of how old you are. It's a key, centralized control mechanism for your entire health span.
And we are moving very rapidly toward being able to manually override that programmed decline.
Using systems that are either these highly balanced pharmaceutical cocktail
or these highly controllable, programmable body factories.
Right.
And that leads to a really provocative thought that builds directly on the DFI source material. The researchers themselves stated that if you could combine full cellular reprogramming back to age 12 and regenerate the thymus, you'd have a pretty fantastic combination.
A fantastic combination.
So since the DFI strategy uses mRNA to deliver specific instructions via the stable liver factory, the question becomes, why stop at immune factors? How far can we push this idea of using transient liver-based factories to deliver any set of useful instructions? Maybe factors designed to correct epigenetic drift in the brain, or cellular senescence in the skin or the muscles.
So we might be entering an era where biological health is just software.
Software that we can regularly update.
A software patch for biological decline. Food for thought indeed. Thank you for diving deep with us.